Welcome to today's lecture on translocations, mechanism and genetic consequences. The main objectives of today's lecture include the understanding of the origin and mechanism involved in translocations, the cytology and breeding behavior of translocation heterozygotes, and the genetic consequences of translocations found in different organisms. During the evolutionary history of organisms, the genomes of organisms are continuously reshaped and rearranged. These rearrangements are brought about by different types of structural changes in chromosomes like deletions, duplications, inversions, and translocations. Translocations, also known as interchanges, are those structural changes in which a segment from one chromosome is transferred to other non-homologous chromosome and vice versa. The net result of this event is transfer of genes from one chromosome to another. Translocations were first reported by Billing in 1925 in Stizolobium deringianum, then again by Blacksley in 1928 in Datura stramonium. Since then, translocations have been frequently found in various plants and animals like Drosophila, humans, Datura, maize, Stradiscantia, Oinothera lamarckiana, etc. Based on the type of transfer of chromosomal segments, we have different types of translocations. Say for example, number one, simple translocation. In this case, a single break occurs in one of the chromosome, releasing a terminal chromosomal fragment. This is followed by transfer of this chromosomal fragment to another non-homologous chromosome. But the problem in this type of translocation is that the other chromosome will not allow fusion of this chromosomal segment because the other chromosome carries telomere, and moreover the chromosome in which break has occurred will be left with a non-telomeric end, so that this chromosome will become unstable, and hence it is believed that this type of translocation hardly exists in nature, although it has been reported by Muller and Painter in 1929. We have another type of translocation known as shift or intercalary translocation. In this case, there are three breaks involved. Two breaks first occur in one chromosome, releasing an intercalary chromosomal segment which is acentric in nature, that is without centromere. Then another one break occurs in another non-homologous chromosome. This is followed by transfer of the eccentric fragment from one chromosome near the break within the other chromosome. Say for example, we have two chromosomes. Chromosome 1 has two breaks releasing an eccentric fragment, and chromosome 2 has a single break. Then the eccentric fragment from chromosome 1 is transferred to near the break in the chromosome 2. This is a unilateral translocation or one-way translocation. This has been found in nature and has been first reported by Bridges in 1923. We have another type of translocation which we call as reciprocal translocation. This is the most widely found type of translocation in nature and has been reported in so many plants and animals. This type of translocation has tremendous significance, evolutionary significance also. Reciprocal translocation means when a segment from one chromosome is transferred to another non-homologous chromosome and another segment from that non-homologous chromosome is transferred to this chromosome. That means two breaks at least are involved, one break in each non-homologous chromosome releasing a terminal telomeric fragment. This is then followed by mutual exchange of these chromosomal fragments between the non-homologous chromosomes. So, the net result is that genes from one chromosome are transferred to another non-homologous chromosome and from another chromosome to this chromosome. Such translocations are therefore also known as interchanges. Moreover, if there are 4 chromosomes involved in translocation in such a way that means that includes 2 homologous pairs, and each chromosome of these 2 homologous pairs carries a translocation, or we can say there is a mutual exchange between both the chromosomes of the 2 homologous pairs, then such an organism carrying this type of reciprocal translocation is known as translocation homozygote. Whereas, if only one of the partner of each homologous pair is involved in mutual exchange of chromosomal segments, so that in each pair one chromosome remains normal and one chromosome is carrying the translocation, such an individual is then called as translocation heterozygote. Translocation heterozygotes have tremendous significance because of their irregular meiotic behavior and as well as gamete formation. Origin of translocations. Translocations occur basically by breakage and reunion mechanism. Therefore, any event which brings about breakage followed by reunion of chromosomal segments between non-homologous chromosomes brings about translocation. There are different ways by which these breaks occur. For example, mechanical shear. During prophase or interphase stages, chromosomes are entangled with each other. Therefore, sometimes chromosomal breaks occur. This is then followed by exchange of chromosomal segments. Sometimes interlocked bivalents are formed. That means two different bivalents become interlocked with each other, and during their separation at anaphase, breakage occurs, which is followed by then reunion of non-homologous chromosomes leading to translocations. Translocations can also occur due to crossing over events between non-homologous chromosomes. There are sometimes some repetitive DNA elements or the centromeric elements which are homologous between the non-homologous chromosomes. So, crossing over takes place in such duplicated regions or homologous regions, which leads to exchange of chromosomal segments between non-homologous chromosomes. Translocations can also occur due to physical and chemical agents which are either generally found in nature, or we can also use them for induction of translocations. For example, X-rays, gamma rays or other chemical mutagens, they are widely known for creating chromosomal breaks. Once breaks occur, this is then followed by sometimes exchange of chromosomal segments leading to translocations. Cytology of translocation heterozygotes. Translocation homozygotes basically show normal type of pairing and bivalent formation, and hence the segregation of chromosomes followed by the gamete formation is almost normal in case of translocation homozygotes. But in case of translocation heterozygotes, since one of the chromosome is normal in each pair and another chromosome carries the translocated segment, therefore normal pairing of such a translocation heterozygote at the time of meiosis is not possible, and hence all the 4 chromosomes involved in this, that means 2 pairs of homologous chromosomes which are involved in translocation, they pair in such a way that at Pachytian stage a cross-shaped figure or a plus-shaped figure is formed. That means 4 arms are formed due to cross-shaped pairing of the quadrivalent. Then, depending upon the nature of crossing over in the 4 arms of this quadrivalent and the chiasma formation in the 4 arms of this quadrivalent, as far as its constitution or the configuration of this quadrivalent will be different depending upon type of the cell in which the crossing over has taken place. For example, we can have a complete ring quadrivalent which has terminal chiasmata between the four arms of this plus shaped figure, or sometimes we can have a chain of 4 chromosomes, that means a rod type of quadrivalent is formed if one of the terminal chiasmata, for example, is broken. Sometimes 2 chiasmata are broken between 2 different arms, then at that time 2 bivalents are formed. Sometimes a chain of 3 chromosomes and a single univalent is formed of this quadrivalent at metaphase stage. However, out of all these constitutions, the ring quadrivalent is one of the most important constitution of the translocation at metaphase stage. In fact, basically, translocation heterozygotes are identified by the presence of translocation ring at metaphase stage, or sometimes also by a plus shaped figure at Pechitine stage. Based on the orientation of this ring quadrivalent at metaphase and followed by the chromosomal segregation towards anaphase, then gamete formation, we have different types of configuration, the orientations as far as this ring quadrivalents are concerned, and based on those orientations, the nature of gametes will be decided later on. For example, we have 3 main types of orientations as far as metaphase stage of translocation ring is concerned. First one, alternate disjunction. In this case, alternate chromosomes are oriented towards the same pole, or in other words, the adjacent chromosomes of the translocation ring are oriented towards different poles. Therefore, at the time of anaphase, the chromosomes, normal chromosomes will go to one pole, whereas the translocated chromosomes will go to the other pole. This is followed by gamete formation later on, where 2 types of gametes are formed, one type carrying the normal chromosomes of this quadrivalent, another type carrying translocated chromosomes of this quadrivalent. But since there is no loss or duplication of genes, therefore both these types of gametes are normal, and hence alternate type of disjunction produces all normal gametes. In another type, which we call as adjacent one type of disjunction, the ring quadrivalent of translocation heterozygote is oriented in such a way that adjacent chromosomes with non-homologous centromeres are oriented towards the same pole, so that anaphase segregation followed by gamete formation takes place in such a way that in each type of gamete, translocated chromosomes along with normal chromosomes are present. These translocated chromosomes carry deletions and duplications, and hence all the gametes formed by adjacent one type of disjunction are abnormal, and hence we have tremendous gametic sterility later on we observe. In third type of orientation of this translocation ring, we call it adjacent 2 type. In this type, the translocation ring is oriented in such a way that adjacent chromosomes with homologous centromeres are oriented towards the same pole, and anaphase segregation takes place in such a way followed by gamete formation that the gametes later on carry deletions and duplications, and therefore all the gametes formed by this type of disjunction are also sterile or non-functional due to this type of segregation. Since we have seen that only alternate type of gamete formation will lead to the formation of normal types of gametes or functional gametes, therefore the fertilization will only take place by this type of disjunction, that is the gametes formed by this type of disjunction, alternate type of disjunction. Therefore, when a translocation heterozygote undergoes selfing and produces male and female gametes, it will produce 2 types of gametes on both sides, one type carrying the translocated chromosomes, another type carrying the normal chromosomes, but both types of gametes are functional in both sides, male and female sides. Therefore, random fertilization between these 2 types of gametes, between the males and females, will result into the progeny of translocation homozygotes, translocation heterozygotes, and translocation normal types in the ratio of 1:2 2:1. Robertsonian translocation. We have one characteristic type of translocation. As we have seen before, translocation generally involves transfer of small chromosomal segments between non-homologous chromosomes. Sometimes the whole arms of chromosomes are exchanged or transferred. This type of translocation is called as Robertsonian translocation. For example, we have two telocentric chromosomes, and sometimes these two telocentric chromosomes fuse head to head and result into the formation of a metacentric chromosome. This type of fusion is called as centric fusion, and since two chromosomes form a single metacentric chromosome, this leads to reduction in chromosome number of a cell or an organism in which such a type of translocation will take place. In another situation we call as centric fission. In this case, a metacentric chromosome will break into the centromere and result into the formation of two telocentric chromosomes. In this centric fission type of mechanism, the chromosome number will therefore increase, because from one metacentric chromosome two chromosomes are formed. In short, basically these Robertsonian translocations have tremendous evolutionary significance, because they are responsible for either increase or decrease in chromosome number or the karyotype complement of a species. These Robertsonian translocations can also occur due to crossing over between two acrocentric or telocentric chromosomes. In such a situation, then this will result into the formation of a metacentric chromosome and a small dot-like chromosome. The dot-like chromosome, which is generally heterochromatic in nature, then lost during further cell division, whereas the metacentric chromosome will survive. Genetic consequences of translocations. We have seen that translocations as far as the meiotic behavior of translocation heterozygotes are concerned, we have seen that both adjacent one and adjacent second type of disjunctions results into the formation of defective gametes due to presence of deletions and duplications. So, because of this defect, only the alternate type of disjunction is able to produce functional gametes, and fertilization will take place only such functional gametes. In fact, the net result of translocation, if we see, is the transfer of genes, that is, the genes change their positions from one chromosome to another and vice versa, plus the formation of these defective gametes which result into semi-sterility or sterility of the gametes. These genetic consequences can be broadly classified as, for example, number one, formation of unbalanced gametes and sterility. As we have seen during the meiotic behavior of translocation heterozygotes, unbalanced gametes are formed due to the presence of deletions and duplications during adjacent 1 and adjacent 2 type of disjunctions. Therefore, in a population of translocation heterozygotes, we are expected to see a semi-sterility, that is almost 50% of the population we will find producing semi-sterile gametes, and therefore the overall seed set is low in such a translocation heterozygotes. Then another genetic effect of this genetic consequence of this translocation heterozygosity is, that is the position effect. As we see that the genes basically perform their normal functions with respect to their fixed positions, because various allelic and non-allelic interactions exist with the help of which these genes perform their functions. So, when during translocation these chromosomal segments are transferred from one chromosome to another, this means that the genes have changed their position from one chromosome to another and vice versa. Sometimes this affects the phenotype of an organism, and any such effect in the phenotype of an organism which is due to the change in position of the genes from one chromosome to another, we call it as position effect. Moreover, sometimes breaks occur within the structural part of the gene during translocations or within the regulatory elements of the gene during the translocation process. Therefore, all such genes which have received such breaks will be defective and non-functional, and hence the phenotype of organism is later affected. Then another type of genetic consequence is change in number and karyotype evolution, that is change in chromosome number and karyotype evolution. As we have seen during Robertsonian translocations, the metacentric chromosome breaks, gives rise to two chromosomes by a process of centric fission, and two telocentric chromosomes fuse to form a single metacentric chromosome by centric fusion mechanism. Both these events lead to change in chromosome number. Centric fission leads to increase in chromosome number, centric fusion leads to decrease in chromosome number. Therefore, this leads to change in karyotype composition of a species. For example, we have one very important example in mouse. Mus musculus is one species of mouse which carries 2n is equal to 40 chromosomes, and all these 40 chromosomes are acrocentric or telocentric in nature. We have another species of mouse known as Mus poschiavenus, which has 2n is equal to 26, and in this species there are 14 metacentric chromosomes. It is believed that either Mus musculus has originated from Mus poschiavenus by the centric fission mechanism, that is the metacentric chromosomes have broken near the centromere into telocentricus, resulting into increase in chromosome number, giving rise to Mus musculus, or the reverse has taken place, that is the telocentricus of Mus musculus have fused together to produce 14 metacentric chromosomes, and a new species has evolved known as Mus poschiavenus. Therefore, there are so many other examples where the karyotype complements have changed due to these Robertsonian translocations. Then we have another genetic consequence of these translocations, which are of course a part of position effect, particularly as far as genetic disorders in humans found due to translocations. There are certain examples where translocation of chromosomal segments between non-homologous chromosomes have resulted into very serious genetic disorders in humans. For example, we have one very important example. There was a deletion from chromosome number 22 reported by P.C. Powell and D.A. Hungerford in Philadelphia. This chromosome was in fact later on called as Philadelphia chromosome, and this deletion has resulted into a genetic disorder known as chronic myelocytic leukemia in humans. Later on, basically, it was found that this chronic myelocytic leukemia was not in fact by the deletion only, but it was by the translocation of this deleted segment from chromosome number 22 to chromosome number 9. Translocation has taken place, resulting into this chronic myelocytic leukemia. We have another very important genetic disorder in humans known as familial Down syndrome. This familial Down syndrome was reported to be due to translocation of a major part of chromosome number 21 to chromosome number 14. That means one arm of the chromosome number 21 has transferred on to chromosome number 14, leading to trisomic condition. Individuals carrying this type of translocation are trisomic for chromosome number 21, that is this part of chromosome number 21, and hence they produce characteristics of Down syndrome. In conclusion, we have seen that as far as translocations are concerned, these are the structural changes which involve exchange of or transfer of chromosomal segments between non-homologous chromosomes. The net result of this translocation is transfer of chromosomal segments or genes from one chromosome to another. Then another consequence of this translocation is the irregular meiotic behavior, where only alternate type of disjunction will result into the formation of functional gametes, Robertsonian translocation results into the transfer of whole chromosome arms, and the translocated segments cause position effect, that is various genetic disorders are observed due to translocations between non-homologous chromosomes, particularly in humans. Thank you very much.